Yang, are you there?
Yeah. Can you hear me?
Yeah.
Okay. Hi, everyone. I am Yang Li. I'm a postdoc in Dr. Ma's lab. I graduated at the end of last year in mathematics, and my major research directions include the integration of single-cell RNA-Seq and single-cell ETEC data, and the clustering analysis of the single-cell RNA-Seq data. And I'm glad to have this opportunity to communicate with each other. And that's all. Thank you.
Okay. Thank you, Yang. Thank you, Anjin. Hi, Rusty. I think the new members has introduced themselves. Shall we get started or wait for several minutes?
No, I think we can get started. So, Noreen, have you started recording yet?
Yes, I have started recording.
So we are good to go.
Okay. So hopefully everyone can see the slides on the screen with the full screen. And if you are just joined the class, please aware that we are more freestyle, and you can stop me, you can ask questions, you can throw out your comments, and just feel free. In the first session of this afternoon, we introduced bioinformatics and the big data. After break, we intended to introduce Seurat. This is how things will go. I will introduce some technology background. Only the history level, not the technical level. And after that, I will maybe use about 10 minutes. And after that, Yuzhou, who is my student, and he will introduce something regarding Seurat. And I will still be here, and we can discuss any questions. First of all, sequencing history. I think the DNA sequencing, RNA sequencing, anything gets started from these two gentlemen, Watson and Crick. And they get started. They opened a door to the new field. And another name I want to mention along this not very long history is Fred Sanger. If you know, there's a Sanger sequencing center in England. So this is a Sanger. Currently the Sanger sequencing center is still the very powerful and a leading position in the whole world. And since 1950 to right now, a lot of sequencing technology has been generated. We call them first generation, like the green parts in this timeline, the second generation, like the yellow parts, and the third generation sequencing. Or people just call next generation sequencing for all. And some names you may be familiar with, like 454 sequencing, like Illumina. I think Illumina is quite popular. I will go into some details regarding Illumina in my following slides. But there are some very popular names, like nanopore sequencing, which belongs to the third generation, using which we can generate a long reads. That's somehow very important to some critical biological questions, like how to infer the alternative splicing. If we have a long reads, that the transcript assembly alternative splicing can be more easily to be tackled. Okay. And the history of Illumina, like I said, Illumina, this is a very successful company, and along with the history of Illumina, I put a chart showing the cost per genome along the time, since 2001 to 2019, last year. As you can see, there is a starting point of the sequencing technology at the beginning of 2001. Actually, this is all get started from announce from our president, Bill Clinton. He announced that we complete completed sequenced the entire human genome in June 2000. And after that, a lot of sequencing companies and the sequencing center technologies came out. Get started from this time. And if you can check the chart, x-axis represents the time and the y-axis represents the US dollars. At the very beginning, if we want to sequence a whole genome, the cost is going to be $100 million. Yeah, that's a huge number. Yeah, and along the time, the price is going down, but not that fast. The real turning point is 2007. Yeah, what happened in 2007? Yeah, Illumina announced a new technology, so-called Solexia. Yeah, I'm not sure whether my pronunciation is correct for this word, Solexia. But after that, it's a turning point. The price of the sequence per genome is going down dramatically. Yeah, until recently, if you want to sequence one genome, I'm saying one human genome, it's gonna cost you $1,000. That means you're gonna guarantee a very good depth. Currently, if you can sequence more samples, there will discount. I think the price of sequencing one genome can as low as $300. Yeah, from $100 million to $300. So that's the history of the genome sequencing. And a very interesting story I'm gonna share. And like I said, my supervisor, postdoc and PhD supervisor, he got started doing bioinformatics in 1990s. So one day in early 2000, he told us he was invited by a very strange company named Illumina. By then it's a very small company. Invited by that company to give a talk regarding human genomics, regarding the genomic data. And after his presentation in Illumina, after he came back, he immediately tell his wife to buy the stocks of this company. He said, this is gonna be a huge thing in the future. Yeah, and after 10 years, everybody know Illumina. Yeah, and their stocks is going up, dramatically. So that's a technology. That's the power of technology. Can make a bundle even. Okay. If we want to see some real sequencers, there are some pictures. I visited IUPUI and last year, and this is the picture we took together with their director, Dr. Yunlong Liu, and the latest version of NovaSeq 6000. Yeah, I think this is how big of this machine. It will cost $1 to $1.5 million, per machine. Yeah, and the later one on the left side is HiSeq 4000. A lot of data were sequenced by HiSeq, and NovaSeq is more powerful and in technology level more accurate. But either of the HiSeq 4000 and the NovaSeq 6000 are very accessible if your data are generated by those sequencers. Okay. Now I give an example. Yeah, I like this example. Yeah, very much. I know some students are still very confused of what is sequencing data. Yeah, looks like. Before jumping into details, I gave you this example. So Rusty, may I ask you a quick question? Can you figure out which is turkey and which is chicken on the slide?
Of course I can.
Yeah. So my question is not finished, and can you tell me which is chicken and which is turkey?
I have a good guess.
Yeah, maybe you can guess based on color.
Yeah, the top is probably turkey breast, and the bottom is a bunch of chicken breasts.
Okay, good job. And can you tell me which is chicken, which is turkey?
Yeah, there you go. Now you're getting a little. You got other things in there, right?
Yes, there are noise Yeah, there are a lot of other informations, but this is basically the thing we are talking about. When we talk about sequencing data,
I'm leaning towards Turkey being on the right.
Thank you. Yeah, thank you for answering my questions. Yeah, the information I want to deliver is when we talk sequencing data, it's not like the turkey and the chicken on the screen. What we received is short reads, a lot of short reads with noise, with sequencing error, with library preparation error, with batch effect, with a lot of other information within the data. And there are a lot of short reads, yeah, mixed together. Yeah, if I'm saying, hey, tell me the interpretation from these two pictures, it's very, very easy to make an interpretation, to analyze this data. But when you go into the sequencing data level, I tell you there will be some insight, like you can interpret which is chicken and which is turkey. I tell you there are some information encoded, hidden in the data, but that's not a naive job. Yeah, we need some computational methods. We need some computational tools. We need some advanced technology to figure out which is chicken, which is turkey from sequencing data. We need those technology to figure out which is cancer, yeah, which is a tumor, which is normal. We need to figure out which is oncogene, which is not. If we need to figure out which are the important pathways, which is the key regulators, which are the drug target from sequencing data. Yeah, but that's not naive. We need some technologies, and I think the main purpose of this two-week short teaching module is to let you know what kind of well-established and what is available things in the public domain to make insight, to derive insight, to derive knowledge from those big data, from those high throughput sequencing data. Yeah, and I hope the student, even especially the undergraduate student, even you are not you do not have any experience in sequencing data, but at least you can have a sense what the sequencing data looks like, yeah, compared to this original phenotype. Yeah. Okay. I will finish with this slide, and then Yuzhou gonna jump in. Another quote from Temple Smith. I personally like this statement, and I like this future very much. Like physics, where general rules and laws are taught at the start, biology will surely be presented to future generations of students as a set of basic systems. Now we are using our computational techniques, try to infer the complex biological systems. After that, we will try to deliver some rules, principles, some knowledge from the whole system, and then hopefully one day we can teach biology like physics. Okay, and I think it's the time to ask questions. And if you have any questions, feel free to ask. And then Yuzhou, you can prepare sharing your slides regarding Seurat. Yeah.
Okay, you can close your screen. I can share mine.
Yeah. Any questions?
I think we need to hear some questions from the students. So
yeah,
Dwayne, Kayla, Miriam, and Sally, what questions do you have?
Any kind of questions are welcome.
If they don't have any questions, I think we could just give them an exam. They should pass it, right?
Yeah. I didn't say yes to this question. I didn't say yes.
Do you have something? I saw your microphone pop on.
I was just going to say I have nothing at the moment.
Yeah, yeah. And we try to be very layperson. I do believe you will have questions in the following slides, but it's good to be clear, and it's good to keep everybody on the same page at the very beginning. Okay, Rusty, maybe we can go ahead, and when we close to the end of the day, we can leave 10 minutes or so to discuss, to solve questions. How about that?
Yeah, that sounds good.
Okay. Joe, you are free to go, I think.
Okay. So it's my time. Can everybody see my screen with only one whole part?
Yeah.
Okay. So good afternoon, everyone. I'm Yu Zhou, and the second PhD student in the Chin Lab. And the following things I'm going to introduce is a single-cell analysis workshop carried out by the Seurat. So the main topic should focus on this four themes, these five themes. Maybe today I'm going to finish into here Seurat package background, and maybe tomorrow I will formally start to introduce the Seurat workshop and some single-cell sequencing technology. So the first thing I'm going to introduce a sequencing introduction. I will give some example of how we generate single-cell data. Okay. So actually, the single-cell sequencing consists of these three parts. First, we isolate the single cell, and then we prepare the library. And after we prepare the library, we throw this library to the sequencers, and we got the raw sequencing data. And after we process those raw data with some very front-end software, and we can get some well-designed matrix, matrix for the analysis. And before I'm going to the details, I'm going to introduce this background again. So at the top figure is list of almost the technology to separate the single cell. The first is use the peptide to manually separate the single cell into the single tube. And the second, we can use the 93 well plate to by using some sorting machine to isolate single cell into this 96 plate. And then the following five technology is more high advanced because it use machines and based on the microfluidic, which is most popular technology to generate single cell sequencing data. And today we will focus on these two technologies, which one is SmartSeq2 and another is 10x Genomics. These two technology is really representative technology, outstanding technology for current research field, because most of the paper rely on these two technologies. And the 10x is the commercial platform for providing large-scale sequencing. And here, the top figure, top dynamic motion here is the motion under the microscope of how the 10x genomics work. Okay, so let's go to the detail. So basically, the single cell isolation technology can be categorized into the two types. The first, capture the single cell into the microchamber. For example, before we get isolate this single cell, we need to isolate the tissue from the live animal. And if we want to went through the bulk RNA-Seq, we can just ignore the following steps just to follow the protocol and build the library based on the tissue level. And if we want to run the single cell sequencing, we need to use enzyme to digest those tissues and separate them into the single cell. And then we use this different technologies. The first, manually separation. Use the peptides to separate single cell into this microtube, or we use the sequencing, the sorting machine to sort those single cell into the plate, or use another commercial platform called Fluidigm C1, which can sort the cell into this single 96 plate, or we can use another high advanced technology. It's called capture in the micro droplets. And this technology is first developed by the Markov lab I will introduce later. And they developed a technology called the DropSeq. And this figure is downloaded from their, this dynamic motion figure is downloaded from their website. And because remember, we already got this single cell suspension, and we load this single cell suspension into this chip and insert the chip into the 10x machines, and the process will go through. Will go through. And inside the machines, the microstructure of the chip, the working chip is here. There are 3 streams of the fluid. The first stream contains the barcode bits and the up to strings corresponding to this channel is mixed with cell. And here is oil, and this is third channel oil. It's like a knife to cut off this water into the small droplets. And here the channel output from the end is the droplets. And the speed of this process very quick is this dynamic motion captured by the ultra high speed camera. Actually, the speed of generating these droplets is about 10,000 per second. Yes, you hear the right things. Yeah, it's 10,000 droplets generates per second.
Wow, good to know. Yeah, it's first time I heard about the detail.
It's very fast. The speed is very fast.
So that they can capture only one cell in the process, right?
Yeah. Theoretically, they should capture each droplet should contain one cell, but during some bias, the capture rate of this 10x is about 65%.
Okay.
So that means the rest of 35% droplets won't contain cell or contains more than one cell. So the successful capture rate is 65%.
Cool.
Okay, so after we got through separated cell, we will break those cell memory out. We let the RNA released from those either from the macro chamber or from the macro droplets, and we will add the small adapter. Is a kind of a molecular modification on this captured molecule and from the library. And after we got this library, we can upload this library into the flow cell. The flow cell is the chip of the sequencer. All the samples will run through this flow cell, and the sequencers will capture the signal of this each molecule. And the raw data, the row of the raw data of the sequences output is image data. Take example of this figure. For example, if we have messenger RNA only have this 5 nucleotides, and we will have the 5 image information circled by this black circle. The first run is well determined. The machine will capture the figure, and this spot will marked as will light on the yellow color, and the yellow represents the nucleotide A. And the second run, it will capture the blue spot, and blue spots and represents the G, and you got the idea. And we got a lot, and we got the rest of the spots and rest of the pictures, and we got the sequence. The sequence is the AGCCT. So that is the row of the raw data output from the sequencer. And because our computer cannot accept those images, so we need to convert those images into the sequence which computer can recognize. We need to convert those images into this ATCG sequence. So that get involved into the very front analysis is called alignment. We align. Sorry, it's not called. It's called demultiplex. So that means we need to convert those images into this sequence, and then we use this sequence to align to the genome map. So this process we call alignment. And according to alignment, we can generate this count matrix. For this for the student who major in computer science, this matrix may be weird because usually the features list in the column and the observations lie on the row. But in the single cell, we use this like the transpose matrix. Each column represents each cell. Sometimes we call this observation. And each row represents features. In this case, we named this gene. So the UMICons matrix is the output, is an input of Seurat package. Okay, so let's get to.
Yizhou,
okay.
I think we may have a pause here in case there are some questions.
Okay.
I feel you delivered some very technical information. Hi, Rusty, do you think that's a good opportunity to ask a student whether they have questions?
Yeah, good. It's a good time to ask questions because I think, and this is a nice job of visualizing what actually happens. And I think that's one thing that's lost, for particularly with computer science, is, as you said earlier from ending your last session, of understanding how the data was collected also helps you understand the validity of the data. So it's not just taking sequence as letters ACGT. You also understand how it was generated, I think is an important part of that as well, because I think there's also error rates that come along with this. And sometimes there are variants in the sequence. They could just be due to error rate. So so we'll open this up to the students to ask some questions about data collection in this way and the algorithms that were written to maybe convert and what are the error rates.
Yeah, because I only briefly introduced those technology. Actually, if I introduce this in detail, it may take the whole day because it's too much detail.
It's okay. It's okay. Yeah, we just deliver the key information regarding sequencing, regarding how those data are generated from the single cell. Yeah. Until we have the so-called gene cell expression matrix. Yeah, a lot of things can be done based on the matrix on the right upper corner. But before this matrix, like Rusty just commented, there are a lot of things happened. Yeah.
We have a question from Miriam.
Sure.
Hello, I sort of agree with Dr. Gonzer regarding about the error rates.
Yes.
Is there like a high error rate from the barcode itself, since it's like keep on random, like being on random on the different RNA-Seq?
You mean the barcode for the droplet strategy, these things?
Yes, correct.
Okay, you ask a very high advanced question. The quickly answer is yes, it has probability to generate the wrong barcode, but it's not very high possibility. It's very low possibility because the company, the barcode provided by the company, and the before those barcode base coming out from a company, they through a very restrictive quality control process, so that barcode had remained very high consistency sequence. I think the major error noise will be introduced is by the sequencing this step, because the sequencer sequence the each nucleotides, and and those nucleotides have some possibility to read the wrong one. For example, if the real nucleotide for this first one is A, but the sequencers has possibility to read it as another type of, for example, has read as the G by mistake. So I think this is major error generate steps, I think, in the front steps.
Yeah, I will have a comment regarding this question and answer. So the error rate is definitely something very important, yeah, and directly determine how will the data play the role in the folding up analysis. but it's kind of, I mean, it's not necessary, but it's kind of, this part will majority be taken care of the sequencer center or sequencing company. They keep developing advanced technology, try to decrease the error rate. Yeah, every time you receive the data from the sequencing center or company, they will automatically generate the data along with the quality check report. You can easily see the data quality, the error rate within their report. I'm gonna say the space remained for computational biologists in targeting this typical problem is getting small. And a lot of companies, they are designing things from there. But again, this is a very important topic. Yeah.
Thank you.
Okay.
Yeah.
Okay.
Any other questions?
If no question, I will go ahead.
Yeah. So I would like to give another comments to my student and the other student in this class. So whenever, like I mentioned in bioinformatics, nobody can know everything. Yeah. So we keep learning. So the suggestion is so never leave a black box in your mind, in your knowledge. Like I'm not trained in sequencing technology, but I can learn this from Yuzhou. Yeah, I asked him a lot of questions regarding the procedure. I will ask how those colors mean, how those colors translated to letters. That will help help us understand the whole procedure. Another example is machine learning and deep learning. A lot of people recognize in the machine learning and deep learning like a black box. So my suggestion is do not do that. You try to open that black box. There's nothing a human being cannot learn. Yeah, we just learn step by step, and when you change every single black box in a white box, then I think your research will be there. Yeah, that's a quick comment. Yeah, you can go ahead.
Okay, so the next, let's go to the main theme. What is threat? But before we introduce the what is threat, I'm going to know. It's good to know who developed the threat. Okay, these people is really a superstar in the bioinformatics field because he developed this package. He named it Seurat-alike, and so far he got the almost 20,000 citations number, and you can see after the 2015, the citation number increased exponentially. The reason is that before the 2015, he don't have the Seurat as the very famous package. But after the 2015, everyone knows the Seurat and the citation increased dramatically. And he trained in the biology. Actually, he got the dual degree in the Duke University. One is biology, another is music. Very, very interesting. And after he graduated from the Duke, and he joined the Oxford University and trained as a statistician. That gives another information because some students here is from the biology. Maybe you have considered whether we can go to the bioinformatics field because a lot of mathematics to learn, a lot of computational skills to learn and knowledge to learn. But don't worry, you see, here is very successful example of the biology background people go to the bioinformatics. So if you clear your goal and keep studying, one day we will go there. And this is my, and these people also always inspire me. And then we go to the Seurat package. So what is the Seurat? The Seurat is the single-cell RNA-seq package designed for the following analysis, including the general analysis. This part is the workshop, the workshop main theme. I will introduce this part mostly, and the other sub function also important. For example, the integration. because in these days, recently more and more people think one samples cannot satisfy the research purpose, so they want to generate more data or use public available data. So to avoid those confounder bias, they use the integration to remove those confounders. And for the others, they are also important. Spatial information is the new product of 10x. It can simultaneously generate spatial information and transcriptomic information. So this technology developed, I think it's the late last year, and the Seurat group fastly developed a corresponding pipeline for this technology. So we can see the attitude of the Seurat the Seurat group is very admiring, because he always provides very pioneer pipeline for the highly advanced technology. And goes to the detail. What is Seurat? Actually, it's the R package for the IRIS-EDA sequence analysis. It's including the quality control part. For example, here are the 3 matrices for quality control, and do the integration, as I mentioned previously. For the different data sets or the omics data, it can integrate and remove the confounder effect and do the folder analysis. For example, here we can identify which gene is highly variable. And I will introduce this figure later in detail, and I will lead you to generate this figure hand by hand, step by step. So don't worry about that.
Hi, Yuzhou, a quick question.
Okay.
In the three variant plot of the quality control, what those dots mean? Can you give us a quick introduction?
Okay. Each dot represents a cell. That means this cell, for example, we pick this cell. This cell can be detected as 2,500 features. That means this cell can be detect 2,500 genes. And each block dots in this figure represents one individual cells. And this y-axis is number of feature, and this says number of counts. There's 2 different things. Number of counts is total feature number, and the feature means is only 1 gene. And this the last figure means percentage of mitochondrial gene is important for the quality control because we have some assumptions I will introduce later. And the y-axis is percentage of those mitochondrial genes.
So if we observe some mitochondrial genes are very high, so do we need to remove them, or?
Yes. If we observe that some cells express highly percentage of mitochondrial gene, that means this mitochondrial gene that cells has died or been dying. The mitochondrial gene can reflect the viability of the cell.
Okay, good to know. Thank you.
Okay, so anyone has questions regarding this part? Okay, if no question, I'll move forward. So this is the previous introduced is the Seurat-alike package, and the next I'm going to introduce some websites which built in the R-Shiny app. Because from the Chin's lecture previously, I know a lot of students can use R. And the purpose of this slide is to introduce the student who don't know R. And if the student want to analyze the data by using the Seurat, this source is good way to perform analysis. This website can provide the whole pipeline of the Seurat, including the input data and quality control and other visualization and marker gene found and marker gene identification. Here is some a screenshot of this website. And for the quality control, we can adjust some thresholds to make sure which cell should include into following analysis and which cell should exclude. And here is a result table for the differential expression markers, differential expression genes. And if you are a Python user, There is another package. It's very similar to a Seurat, but it's not developed by the Seurat group, by Seurat lab. This package called SciPy. And it also includes the information of analysis, for example, the quality control and data preprocessing, and some visualization and further analysis. The unique feature of this package can do that Seurat cannot do is pseudotime, because the Seurat cannot do the pseudotime analysis. The pseudotime means can use algorithm to decide, because we use the single cell data, right? And those single cells potentially have some lineage underlying their development. So the pseudotime is the technology, the machine, the computer can map those cells into the lineage manner. So that the computer can tell you which cell coming first and can develop or differentiation to another cell type. So that is what pseudotime do. So because the Seurat is the open source package, it hosts on the GitHub. It's started by the 800 people, and you can see the fork means how many people co-developed this package. There are a lot of people contribute to this package, and the issue means how many users use the Seurat package, and they make some questions and issues so that they post the issues and questions on the GitHub, and the Seurat group will respond immediately and fix those questions and develop the Seurat package more robust. The current version of Seurat is 3.1, but it went through a lot of previous versions. And this timeline is showing the developed history of Seurat. In the 2015, they developed the first version of Seurat. And this paper published in the Nature Biotechnology, which has very high impact factor. It's almost 40, I think. And so far, it got 1,300 citation numbers. And the second version of the Seurat is published in Cell, but this paper is emphasize the dropsticks as not this technology, not the Seurat package, but but in this Seurat version, they form a very mature package and host on GitHub. And this paper published in Cell, and till now it got almost 3,000 citations. And this true update is minor updates regarding some visualization or the documentation for the Seurat package. And through the 2017, the Statista published the second version. This version, they use the integration technologies so that we can use different experiment results and integrate them together and to do the further analysis. This paper published in Nature Biotechnology, and so far it's got almost 2,000 citations. And the latest version is the Seurat 3.1, and this based on the version 3.0. And this version also emphasizes integration, but the difference between the previous ones is this integration integrates the different multi-omics data. For example, it can integrate some spatial or chromatin accessibility. I will introduce late how it works. But you can know the whole timeline is very. You can see it's like the very well designed package. And it's also one of my goals. If I can publish our package like this, I will maintain it until everyone can use it.
So I like this idea. I like this idea. Yeah.
Okay. Okay. So let's go to the Seurat 1. Why we use it? Because the previous the Seurat 1 emphasizes the spatial information with the messenger expression level. In development biology, the single cell are everywhere, but do not contain the special information. But can we add the special information on this single-cell RNA-seq? The answer is yes, because this group, these two people are very big guy in their field. For example, the Alexander was the researcher regarding the zebrafish. So they provide zebrafish embryo for the single-cell RNA data source. And I've averaged she is very, very important guy that I want to introduce today, because she is the boss of the Satija. In this time, Satija was a postdoc of her lab, and she was a computational biologist, and she have a lot of publications regarding the cancers and regarding the modeling, and statistical number is almost 70,000. And recently, actually in May this year, she left the Broad Institute and she joined the company called GeneTech, which will focus more focus on the industrial field. I believe after she joined that company, and the company will develop very well. So personally, I recommend you should buy the stock of that company. Okay, so the Seurat-1 deliver information of spatial information and the expression information. Here is the embryo, zebrafish embryo, the structure. Each this animal ventral or this four corners are the coordinates of the zebrafish embryo, and they have hypothesis. The different locations of the cell in the different regions will express the different expression gene, and the result is yes. From this figure, we can see the gene X here is the upper sphere of this zebrafish embryo, highly expressed in this region. You can see the histogram here that is highly expressed by the gene X, and the gene Y at the top layer, they highly expressed as the animal this layer, and the gene Z highly expressed as the dorsal this part. Along with this package, along with this paper, Satija, as the first author, developed a pipeline. It's the first version, Seurat, but it's not major. It's a prototype. It's not a major package because all the code are compressed in zip cell, and this zip file are uploaded as the supplementary of the figure. So it's not mature R package. So to the Seurat 1.2.
Hi, Joe.
Yes.
Yeah, may I have a quick question?
Yeah.
So we know there are multiple versions regarding Seurat, like 1.1, 2, and to 3.1. So are all the previous functionalities included in the latest version, or we need to maintain different versions?
Some functions they delete, and some functions they improved. So okay. My answer is they delete some function and also includes the old version functions.
Okay, so which means we can just use the latest version for Seurat.
Yes, we can use. But yes, and if anyone want to use old version, the Seurat official website also provides a link. You can download the old version of Seurat package.
Okay, because some algorithms are slightly different between different versions, right?
Yes. Yeah.
Yeah. If they want to reproduce something using the exactly same version, they are free to download the previous versions, right?
Yes.
Yeah. So here I have another quick question to Rusty. So I think in our slides we have some code. So how should we, teach that part? Should we expect every single student in this virtual room to really implement that on our class, Rusty?
With the code?
Yeah.
And have we seen the slides with the code for sure yet, or are they coming up?
They're coming up, not today.
The next class.
Next, maybe tomorrow. When we have those code, I mean, which one do you prefer? Let the student implement run the code simultaneously during the class, or we can share the code in once they can do that, offline. Which one do you prefer?
So what I would suggest is share the code so they can play with it a little bit tonight to get familiar with it. Mm-hmm. And then tomorrow they're able to run simultaneously with you as you're doing activities.
Okay.
That seems to me seems to be more active learning that way. Would all the students agree with that?
Yeah, that sounds perfectly good.
Yeah, I have a script, so I can send them to you.
Yeah, regarding sharing the documentation, the slides and the code, Rusty, how to proceed? Should we send along with email attachment or other ways?
I think Noreen said we've used Dropbox links before, but if you want to send the email to Noreen, she can distribute it as well, whichever is most convenient at this point.
Okay. Then I will share my slides and Yuzhou's slides, including the code after today's teaching, and then so that the student can have a sense what kind of code they need to run tomorrow afternoon. And Noreen, can you send me a quick email, and then I will share the information with you?
Sure.
Is that okay?
Yeah,
okay.
That's fine. That's totally fine.
And so therefore, students, you have an assignment tonight to go in and look at the code and see your knowledge of R and what is written there and how that code looks, and see if you can predict what some of the things do so you're familiar with it, that we can have an engaging conversation tomorrow.
Yes. And
absolutely.
I will suggest regarding the introduction part, we can roughly stop here because it's almost done. And can you quickly move to one slide regarding having the code?
Yeah, that's why.
Just one example. Maybe a real data analysis. Yeah.
Yes, actually, it's one part of the whole code. I've totally prepared the eight sections for practice, and if allowed, I will send those 8 sections in one script to every student to practice tonight. But at least today, but at least tonight, every student should install the Seurat package by following the 1.1 and 1.2 because it's really important for the following tutorials.
Mm-hmm. So can you move to the next one with a real data analysis page?
Oh, I didn't include the following because I plan to stop here. Yeah.
Okay, but I will suggest you share those slides together with the student tonight, and so that they can see what kind of functionalities, what kind of code. So Rusty, so basically we will share a slide. Maybe the upper is a code, the bottom are the visualizations, the results, or very similar to RStudio style. And we really hope students can check those code and results and really type in those command lines on your computer, everything in R. So don't worry about the environment. And if tomorrow afternoon, anybody have any specific questions, Yuzhou and Cankun and other students should be here for consulting. Yeah, but we really hope every student can at least run some Seurat analysis showcased on this class. Yeah, but other students, don't worry, we do not have exam. Yeah, just explore for fun. Yeah, just explore the functionalities provided by Seurat. And learn something. That's the important thing. Rusty, I think we still have several minutes for questions.
Yeah, I think we're good to open the floor for questions. And particularly for any students who want clarification, what they're supposed to do tonight to prepare for tomorrow, etc.
Mm-hmm.
Are you guys
So we're
going to make me
supposed
call out?
I do have a question. So we're supposed to just reproduce like the visuals that you had on the slides.
Yes, I think you will. Yeah, you will see how to generate those visualizations showcased on previous slides. Those beautiful cell clustering, those beautiful dimension reduction results, the trajectory. I think
Yeah, you will generate those figures step by step.
Yeah.
Okay.
Yeah, and the beauty is after the class you can you can clarify, I'm the expert in single cell RNA-seq data analysis.
Okay, thank you.
Yeah.
So, any other questions?
Kayla, Miriam, or Sally?
So do we need to replicate version 3 of Seurat or version 1 of Seurat?
You know which version do you prefer?
I prefer you should download the latest version 3.1 if you directly use this command. I will show here. If you directly use install package Seurat-alike host on the screen, you will automatically download the latest version.
Yeah.
And to be noticed is the recommends the R environment is the R version should greater than 3.4. Please notice that.
Yeah, that's cool.
Thank you.
Yeah, today just a warm up. Real fun tomorrow.
Tomorrow, yeah, tomorrow sounds like
practice.
the fun
Yeah.
starts tomorrow.
Yeah.
So
Kayla,
quick
you were saying
question.
something?
Does it matter if anyone's running R4 versus 3 point whatever?
I don't know. I don't know how exactly that version will affect things. Usually, it's only a minor minor portion of the functionalities, but to make sure the reproducibility, like everybody generates exact same results, yeah, we just recommend that you use the latest version Seurat and our version more than 3.4. Yeah.
Okay.
Okay, thank you all for joining us today. And if no more questions, we can see you tomorrow.
Yes, and we'll be setting up. Noreen's going to set up a new meeting request for tomorrow. Is that correct, Noreen?
Yes,
So
I will do it. Yep.
let everyone know that there'll be a new one, and the one you want to join is the one that Noreen organized by Noreen.
Thank you in advance, Noreen.
No problem.
Otherwise, we will see you tomorrow at 1. Is that correct?
Yeah.
1?
Yeah. Enjoy the rest of the day. Bye-bye.
You as
Thank
well.
you.
Thank you, guys.
Okay, thank you.
Thank you so much. Have a nice evening.
See you
Yeah,
tomorrow.
you too. See you.